So, you've been in the space of bioinformatics for any period of time. You've known that there is quite a bit of confusion out there, in that there are many different programs or web applications that someone might need to utilize in order to carry out a specific workflow. Case in point, you can visit or utilize as many as 20 different programs or web applications to go from sequence data to some type of meaningful outcome or generation of new hypotheses. There's an interoperability issue in which when software is designed, it's not necessarily designed with the broader research landscape in mind, meaning that when you analyze a data set, oftentimes a file format that comes from that needs to be imported into the next application downstream, having to deal with file conversions or file management. And this makes sharing data quite difficult. It makes collaboration quite inefficient. As well, you'll find that there's a steep learning curve. Users typically find that there are many different applications out there to choose from, and qualifying which application is the best for their specific purpose can be tricky. Oftentimes, biologists don't have the time or the bandwidth to benchmark algorithms or to compare 8 different software packages with one another to find out what is the most useful, what is easy for them to use, what gets the job done. And the solution to this is if you were able to design a software application where you had all of the existing applications combined underneath one framework, your web services, as well as your analyses in one place, reducing the number of clicks or steps or different places that you have to go to in order to carry out your work. Also, if you could have a software application that was designed to be extensible, in that there's a public API that is made available so that as your research changes or as you need to customize features, you can easily do that. And finally, if you're able to have your software designed or informed through the consultation and collaboration with biologists and bioinformaticians, that is key because you don't have a team of coders locked away in a room developing a piece of software that has not been informed. You want your software to be informed so that that tacit knowledge that the virologist or the epidemiologist have can be used for the design process. And essentially this we did with Geneious. Geneious is a cross-platform Java application modeled on an email client interface. And it's cross-platform, meaning it doesn't matter which operating system you have, whether you're working with Linux or PC or Mac OS, Geneious will run on your machine. And we've taken advantage of something that you already know how to do within Geneious. We've designed Geneious with this email client interface, and that everyone understands how to manipulate their email. You know how to create files, rename folders, color them, drag and drop. For organizational purposes, this is critical. And perhaps one of the most important things with Geneious is that we've integrated industry-leading algorithms, peer-reviewed bioinformatic algorithms, algorithms that have been well-published, well-tested in primary journals, so you know exactly what's happening to your data. The opposite of this approach is the black box approach, in which your data go in, something happens to it, and then you get some result, but you don't necessarily know what's happened to your data. We also support open file formats. We support 60 different file formats, and we've made it very easy to import your data. We have a drag and drop system so that if you don't know what type of file you have, you just drag and drop it into Geneious. And Geneious will tell you what type of file it is, whether it's FASTA or FASTQ or chromatogram. And then we've also organized Geneious into a complete workflow. While we analyze data, and we feel we do this quite well, what happens if you have no data? So we're looking at a more upstream. If you need to go through a discovery phase, we've built Geneious so that you can go out and get your data in a trivial fashion. And then once you've discovered your data and you've analyzed data, now you need to publish it, either for a white paper or for a peer-reviewed journal or a poster, or maybe you just need to print out a chart for your lab notebook. We've made it that a person can do this easily as well. So you don't have to export a figure and then import it in some file editor to adjust colors or spacings or things like this. And with Geneious, we fill a very important niche, especially in these times where funding is a bit strained. Geneious, we're looking at extending bioinformatics resources. So we're not expecting to replace bioinformaticians or computational biologists, but what we are looking to do is we're looking to free up their time so that they can work on harder problems. We're trying to get more and more biologists to pre-process and to analyze their data. As well, with Geneious, you can have a centralized software maintenance schedule. So you don't have to maintain separate copies across an institute or across a site. You can roll out a major version release with a couple clicks of the button. And then we also strive and aim for this collaborative space. So we want to make data management and we want to make collaboration very easy for the user. If you have a central data repository at your institute, we want you to be able to have access to that from your desk. And we want this data management piece to not be as problematic as it has been in the past for biologists. Geneious covers a number of technical features, and I can't talk about all of them today, but I'm happy to take questions after this talk, or I'll show my email address a little bit later. If you have any other specific questions, I'm pleased to answer them. But we work in phylogenetics, of course. That's where we've started. But we're also, we've created tools for next generation sequencing, for sequence alignments, for clonings, as well as visualizations of many different types. And before we get started, I feel we need to have a bit of a tour of the interface. So you go to the website and you download a free copy of Geneious. You install it on your machine. This is what opens up when you launch it. And to the top left, you see this local database, this file directory structure I referred to a little while ago. This is the thing that we built on that email client interface, where you've got your local files and they're named and or local folders. And then within the folders, you have a number of different files therein. Below that, we have a listing of live services, and these are services that are always on, services like NCBI or PFAM, as well as Uniprot. We also have a few services that tie into some of the UPathDB websites, AmoebaDB and PlasmoDB. And this is where you can go to query for a genome, an NCBI, for instance, if you want to pull down E. coli K12. As well as all of the metadata, all of the annotations associated with that sequence data. You can do that from within NCBI. As well, you can, if you have sequences or a genome that you would like to publish to NCBI, you can do that from within Geneious. At the very top, when you click on a folder, the contents of the folder will open up in this document table. In this case, I have a number of different files. I have an Arabidopsis file, I've got a config file, I've got a primer, as well as a multiple sequence alignment. At the very top of the file names, we've got a number of different column headers that you can hide or expose. You can move them around according to what your interests are. And just to the left of that name, we've also created nifty icons. So you can tell immediately what type of file you have. You can look and see that you're looking at a phylogenetic tree or that you're looking at a set of primers. When you click on one of those files in the document viewer, you'll see what that file looks like. So at the top of the document viewer, we have what's called a mini map. So you have a global context of where you're at on the file. And Geneious always opens up to the most macro view so that you can decide where it is you want to drill down into. We also have some annotation information, some directional information. To the right of that options panel, or to the document viewer, you can control what is viewed in the document viewer using this options panel. The coloring of your nucleotides, which annotations you would like to have exposed, and everything in that document viewer you can export as a figure or as some type of graphic, whether it's SVG or JPEG or GIF. We have a number of different options that you can export that document viewer. To the right of the options panel, we have a help and tutorial panel. And this is designed for self-study. If you have 2 or 3 people within your lab and you would like to take an hour or 2 each week to sit down and to go through some exercises, we've made some pretty robust tutorials so that you can understand and get up and running within Geneious. If you have already got a good command of Geneious, you can always collapse this window and you can increase your screen real estate for the other panels that you see. And at the very top, there is the menu bar. And the menu bar is one of the main ways you're going to interact with Geneious. So we have icons set up for sequence searches, for building trees, for performing cloning operations, or as I mentioned earlier, if you'd like to submit some sequences to GenBank, you would use that menu icon up at the top. Now, 2 things I always like to point out is on the right-hand part of the menu bar, there's the backup button. And that green button is very important in that you don't really appreciate the value of backing up your data until your data are gone. And so I highly encourage everyone to perform regular backups of their data. And then as well, just as importantly, there's a support button where if you're having a hard time within Geneious, if you're stuck, or if you're just not quite sure what's happening, you can click on that button and submit an issue, which will get shunted into our support queue. And this support queue is fed to our team in New Zealand, in Auckland, as well as me. I'm based outside of Seattle. And this support button is very handy in that we're able to get to your issue quite quickly. When I first started using Geneious, I was actually a user for 4 years before I jumped on board. The longest I ever had to wait to have an answer for a support issue was about 4 hours, maybe 4.5 hours. And this was something very striking to me. So we're getting rid of that lonely grad student syndrome in the lab. So I mentioned earlier, Geneious handles a lot of different features. We handle multiple sequence alignments, and we have a lot of different options for that. In this case, I've got 20 genes that I have selected within a list. And when you select your gene list and you go up to the top menu bar and click on the alignment icon, you'll have this Geneious dialog window that pops up. And I certainly won't go through all of the different alignment features, but I do want to point out we've got a number that we've implemented. We've implemented the Geneious alignment algorithm, which is a variation on Smith-Waterman. We've implemented the muscle alignment algorithm, and we're working with Robert Edgar for this. We have availability for the Clustal W alignment algorithm, as well as MAFT, which is based on fast Fourier transform. We have made sure to expose all of the different options and parameters that are available in any of these alignments out there. And all of these you can download and you can use separately if you like. But in particular, muscle has a number of different parameters, and we don't hide any of those parameters, so you have command for all of those. We have some new alignment features pushing out for this next major version release, which is dropping on the research community October 8th. We've included LAST-Z, and LAST-Z is a BLAST-like alignment algorithm for pairwise alignment of chromosome-sized nucleotides. We've ported this over from the Linux environment so that you can run this on your Mac or your PC. We have also included the MOVE whole genome aligner too, using the progressive MOVE algorithm. So if you're comparing a number of different Bartonella species or influenza A isolates, you're able to have a quick qualitative view using this MOVE genome alignment. We've also made it at the very bottom of that Geneious dialog box. You can see we've got the reference for where the algorithm was first published. So you can go straight to the publication and you can see all of the different parameters or the different ways that this algorithm was used. We've included Bowtie for client for Geneious R7. And Bowtie 2 is part of the drop-down menu box when you want to do a reference-based assembly. I've got a number of different options shown here, but this is to show that we have also given you options on the bottom left to trim your sequences, as well as to save an assembly report and to save that analysis in a subfolder. So you can compare different settings of your aligner. And here I've got a figure of an Ames Bacillus anthracis contig that I generated. So what I did was I aligned a set of Illumina reads to the strain Ames A. And so this is showing you where the reads pile up, the type of coverage that we have. We're also able to call SNPs to look at coverage a little deeper. And so for next generation sequencing, this is what the view would look like to the user. So we're including the Velvet read assembler for local clients. And here I've got a number of different Illumina reads. I trimmed the 3' end because Illumina data is more error prone on the 3' end. And here what I've got viewed is the sequence that has been trimmed, and it's annotated in that light pink color. And this sequence will be ignored for downstream analyses, but we haven't done away with the sequence altogether, so that if you want to come back and reanalyze your data, you're able to do that. And we've got the reads colored according to quality. We have a number of different cloning tools. Primer design, you can design primers on multiple sequence alignments. You can create custom constructs. We have gateway cloning that we've implemented. We've added a few new cloning features to help the synthetic biology community. We've included a Gibson assembly for Geneious R7. And this is the dialog window that shows up to show you how your Gibson assembly is to be ordered, as well as a Gibson report that is generated, which is very nice, especially if you need to, if you're developing protocols and you need to include this information in your lab notebook. We're including topo cloning for this new major release, and a visual of the topo cloning dialog box. We've allowed for TA cloning, for blunt cloning, as well as directional with overlap. Within the phylogenetic space, we have extended our feature set. So we're not offering the Geneious Tree Builder or Mr. Bayes or PhyML or Popstar. Those we had in this prior version, in R6. We've extended out to include RaxML, Garli, as well as FastTree, so a number of different maximum likelihood methods that you would have the choice of using. And we have our sights set on the microbiome community that are doing very large phylogenetic trees and tree comparisons. In the next generation sequencing space, Geneious is platform agnostic. So whether you're using Illumina or 454 or PacBio data, Geneious will handle your data. And here I've got a visual of contig that was generated, as well as where the paired end reads line up, and coverage graph shown in green. If you want to call SNPs, which are very easy, if you're using an annotated genome, what's nice within Geneious is you can tell Geneious where you'd like to look for the SNPs, whether only inside the CDS or if you're looking for either synonymous or non-synonymous. And you can set your maximum variant p-value threshold. We've made variant calling quite easy. In this visual, I have the variants that were called just below the reference-based assembly, and they're annotated in that orange color. You can change the view so that you can march through or work through your variants to sort your variants. So in this table that I have set up, you can sort your variants based on variant frequency, or if you want to look at strand bias, if you want to look at a particular set of changes, if you just want to look at transversions or insertion deletions, you can sort according to that. And we've made this so that you can throw out your false positives and drill down to the more likely variants. You can also export this as a table to import in downstream tools if you're using R, for instance. Geneious handles chromatogram assembly. I've got just a very small set of fragments, but Sanger sequencing, we still have a number of users who are using Sanger sequencing. And within Geneious, you're able to trim on quality score, or you can trim primer sequences or vector sequences off of your fragments. And here I have an alignment of these fragments as well as the orientation. And you can see on either end, 5', 3' end, that we've got the trimmed regions annotated in pink. We have very robust annotation tools. We have rich copy-paste. If you are looking to do larger scale annotations, we have this workflow builder that we've designed. And we've included a couple of different sample workflows. And the way the workflow builder works, if anyone is familiar with workflows, you know it's very process intensive. And we have Geneious so that you're able to identify each specific step in your workflow, as well as the different parameters of that particular step. And this is very handy because you can share workflows now with your collaborators. You can fork different steps within your workflow. So this is an aim to, again, make your work more efficient, to reduce the number of times you have to perform repetitive tasks. And the old adage is, if you have to click something or do something 3 times in a row, probably you need to look at automating that step. So the latest features we had for R6, we included new locations table view. We included support for read mapping to multiple locations, VCF import, drag and drop imported zip files, as well as a number of different export images like PDF or SVG. We upgraded Primer 3 for all of your cloning processes. So if you're designing primers on multiple sequence alignments, Primer 3 is what underpins that. New features for R7 are those features that I mentioned a little while ago. Gibson, Topo, we're looking at implementing Golden Gate in an upcoming release. We've got the 3 maximum likelihood methods for tree building. We're including LastZ and Bowtie 2 for client. We are in the midst of working on a haplotyping plugin, and we're starting out small with an algal genome, but our aim is to work higher to more complex organisms. We've also built in a feature for codon optimization. The workflow builder that I mentioned just a few moments ago, we are offering. And we've got smart folders, so you can do automatic sequence searching or literature searching against NCBI. If you'd like information, you can go to our website and you can download Geneious, or you can look at tutorials. We're going to have webinars set up there shortly. We also have a very easy way to go and to get whichever version of Geneious you need for your specific machine. One of the benefits of Geneious is that we have a very robust community, and we have a number of people who contribute their requirements or their feature requests, and they do that through this great tool, this Geneious user forum. If you have a problem, I do recommend coming to this Geneious forum first. Chances are the question has already been asked. But this is a very community-rich, a very community-driven approach. And if you have a problem or an issue within Geneious, the beauty of this is that when your question is answered and so that you're satisfied, then you close out the issue, and we continue to work with you until we get your specific question answered. Here's my contact information. You can go to our website. You can certainly email me if you like. We are at a lot of conferences all throughout the year. So if you're at a conference, please take care to look us up. If you do use Genius, please stop by and let us know how you're getting on with it. Or if you have a feature request, we would love to hear about it because this is one of the things that drives our development.